computers to print out that lab tutor?
I know you didn't.
Okay.
Just give me a list of the
So
people.
like tonight, when we do it, we can save it to like a little memory. stick.
Tonight, it's going to be done as a demonstration.
Oh, okay.
Yeah. Well, I'm gonna start walking for
Okay. It's supposed to have 21 people. Okay. We're going to just start on some of the basics of Chapter 13 when we get into more of the blood, the electrochemical or electric potentials of the heart. We'll pick that up on Tuesday. But I just wanted to introduce this. So, Chapter 13. Circulatory system. You have really two types of circulatory. You've got circulatory systems. You've got the blood and you've got the lymphatic. The blood carries oxygen and nutrients to the tissue. The lymphatic drains the tissue and carries it back to, dumps it into the veins and back into the heart and lungs. So it drains the tissue, the fluid from the tissue, interstitial fluid. And Andrea, nobody's sitting down there, is there?
What? Oh, no, I should. Yeah. Trying to figure it out.
Okay. So, I forgot to get my little laser.
Last class, you bring up the stuff.
What's that?
You'll bring it like the last class.
Probably. It'll take me that long to remember it. So you've got here, the heart, you have a portal system where the blood is going to the lungs and back to the heart. So you have a venous blood carrying, which is going to the heart. The definition of venous would be blood that's going back toward the heart. It comes out, but it's the only place that you have oxygenated blood in a venous system. But here's just a diagram. We will certainly get into that in more detail. Okay, what does the blood do? Transportation. Carries gas, oxygen, carries nutrients, glucose. Waste, it picks up carbon dioxide, it picks up BUN, it picks up other waste products from the cells, from the tissue, carries them back, and they're generally going through the liver or the pancreas or the spleen, generally the liver or spleen where the chemical wastes are removed. In the lungs, the CO2 is removed by and exchanged for oxygen. How is this done? When we talked about cellular respiration, we talked about how gas is exchanged from the veins into in the lungs. How's it done? You're down to a one-cell thickness between the epithelial cells. Your epithelial cells in the alveoli are one-cell thick. You've got a red cell coming in with a high concentration of carbon dioxide, and you've got the lung tissue with a high concentration of oxygen. What happens? It diffuses. There is no active transport. It just simply diffuses across. Okay? Also, regulation. Endocrine system. Endocrine dumps the hormone into the blood. The blood carries it to every cell in the body within about 7 seconds. Only the cells that have a receptor can utilize that hormone. But it is part of the regulation. So if a thyroid hormone, if TSH goes to a muscle cell in the big toe, it ain't going to do nothing. It goes to the thyroid, then it's going to get in, it's going to cause the thyroid to produce thyroxine, and it's going to be useful. Protection, clotting, and immune. The blood carries clotting factors. it carries platelets, it carries fibrinogen, it carries other clotting factors. Roman numeral 1 through 13, used in various parts of the system. We will touch on it. We're not going to get too in-depth on it. But remember, when we in the first lecture, the very first lecture, we talked about the clotting system being a positive feedback. That was a long time ago, wasn't it? I'm surprised at my age I even remember what went on back there. But we'll get into it a little bit, but it is a positive feedback. Eventually, obviously, it becomes a negative feedback. But, the cut releases a tissue thrombin, which causes platelets to aggregate, which releases thromboplastin, which causes other clotting factors to activate, which in the end forms a fibrin clot, or fibrin flow. And we'll go through that. Immune, it carries globulins. It carries alpha globulin, beta globulin, gamma globulin to fight disease. Some are produced by the liver, some are produced by the white cells. Your gamma globulins produced by your white blood cells. You have two system components. You have the cardiovascular system. You have the heart that has four chambers, 2 atria, 2 ventricles. You have the blood vessels, arteries, arterioles, going in size down. Arteries, small arteries, arterioles, capillaries. Capillaries, and then to venules, and then to veins, and then back into the heart. Inferior, superior vena cava. Then you also have the lymphatic system. It kind of parallels the circulatory system, but it doesn't have the heart. It drains fluid from the tissue. The lymph nodes kind of purify it, clean it up, get some of the junk, the bacteria and that out. And then it dumps back into the veins, then back into the heart, and then to be circulated again. Organs of the lymphatic system: the spleen, the thymus, the tonsils, and the lymph nodes. In an adult, the spleen is still working, very important. You can certainly live without it. Many people do it. A lot of football injuries, you rupture a spleen, they just go in and take it out. You have a little more trouble with your immune system after that, because it helps, and we'll touch a little bit on it. We'll get into the blood and in later chapters. But it helps the body recognize things as foreign and create antibodies against them to protect the body. The thymus in an adult is pretty much shriveled and useless, unless, like a friend of mine who just had a cancer of it, and so he ended up having to have it out, and it's buried under the breastbone, down by the heart, and they've got to crack the chest to get in and get it. Tonsils, again, in children might have some use. They're pretty much holdover from the caveman days. They don't do much for us except get swollen and cause a lot of trouble, and generally they get taken out. Lymph nodes, again, are in line in the lymphatic system. They tend to grab the particles out, help purify them. You get a cancer. You have a breast cancer. They always are going to take the axillary lymph nodes and the lymph nodes around the breast to see if the cancer cells have gotten into the blood or into the lymph nodes. If you have a cancer, you'll have a diagnosis of cancer in situ, which means it's all in there. Or cancer It just went away. When we get more into the lymph system, I'm sure it'll be back from long-term memory by then. Okay, composition of blood. You have the particulate part of blood and you have the fluid part of blood. You have blood plasma. Plasma is the fluid part of blood. Now, the difference between plasma and serum. Plasma has all the clotting factors. They've been inactivated in some way, that's why you have plasma. But your fibrinogen is there, your clotting factors, your platelets are there, they're not tied up in a clot. Serum, you allow the blood to clot, and then you spin it down. So you have plasma and you have serum. So if you spin it down, what you're going to see is the plasma, which is the fluid. And if you've got a buffy coat this big, you're in trouble. Normally when you spin it down, you don't see a buffy coat. This guy's probably got a white cell count of somewhere around 20 or 30,000, which would put him up into a very bad infection or a leukemic type state. Normally you won't see a buffy coat, but if it is there, that's where you see it. The red cells are heavier because of the hemoglobin in them. So you have the red cells, the buffy coat is white cells, plasma is the fluid that they're floating in. And here, we looked at the red blood cells, the biconcave discs. We looked at white cells when we were doing the microscope. And platelets are the little, they're a tissue unit. They're not a cell of their own. In the bone marrow, you have a cell called a megakaryocyte, and these are actually little bits of cytoplasm off the megakaryocyte. They live in the serum or in the plasma for 4 to 7, 5 to 9 days, something like that. And they all have different functions. We'll get into that. Plasma is the fluid part of the blood. You have plasma proteins. Albumin. We talked about this when we did osmosis across. I told you we're going to get all back to this. We talked about albumin when we did osmosis. Diffusion across the semi-permeable membrane. In your body, the blood and the lymph is full of albumin. The main thing it's there for is to draw tissue out of, draw fluid out of the tissue to maintain that balance. There is a syndrome they call nephrotic syndrome. And in this syndrome, the people cannot filter the albumin out in the kidney, and they just pee it out. And they pee tens or hundreds of grams of albumin out during a day. They do not have the albumin in their system to pull water out of the tissue, to pull fluid out of the tissue. You walk into their room, you guys know what the Michelin Man is? Yes or no? The big, puffy, like the Pillsbury Doughboy. That's what they look like. Because there's no way to get the water out of the tissue, because there's no albumin to draw it out. And these people, we buy the albumin very concentrated, and we just keep pumping it into them. At the same time, you'll give them steroids to help eliminate whatever autoimmune reactions going on in the kidneys so that they start retaining their albumin. And eventually they seem to get better. But it's very, it's kind of disheartening when you see somebody that, for one little thing that's wrong, is that sick.
Is albumin a large molecule?
It's a fairly large protein.
And it needs to be in the circulatory system?
Yes. In the circulatory and in the lymph system.
But if it's out, wouldn't it draw water out with it?
Well, once, if they're peeing off the albumin, they pee a lot, but the water doesn't come out of the tissue. They'll pee what's in the circulatory system, but water won't come out of the tissue into the circulatory system because there's nothing to draw it out. The gradient for diffusion is in, not out. Because the concentration of albumin is, probably 10 times higher in in the circulatory system than it is in the tissue. Because it's really designed just to pull, that's all it's really there for, is to pull water out of the tissue.
What about sodium and glucose, stuff like that? It won't pull it out. You need albumin?
Yeah, albumin's in a much higher concentration. It's in grams. You know, you measure your sodium in milligrams, your glucose in milligrams, thousandths of a gram. You measure your albumin in the serum is somewhere between 3 and 5 grams per 100 mils, whereas your glucose will be 90 milligrams. So you're talking about 1/300ths of it. So it just doesn't do the job. Okay, you got the albumin. It creates osmotic pressure. That's what we've just been talking about. The globulins, they may carry some lipids. Your gamma globulin, made by the white cells, carries the antibodies. This is what they use to fight the infections. Fibrinogen is only about 4% of your protein in the blood, and it forms strands, and it forms plugs over clots, and helps with the blood clotting process. There's a disease that we'll talk about. It's called DIC, disseminated intravascular coagulation. Unless you get it dealt with, it is really, really nasty. Generally, the most time that I've seen it, it's been in women, with maybe a torn placenta previa. The placenta's coming out before it's ready. A lot of the tissue clotting factors released from the placenta, and what essentially happens is you start clotting all through your blood system, your circulatory system. That doesn't kill you. What kills you is that you use up all your clotting factors and you bleed to death. It's disseminated because it's throughout the body. It's intravascular because you're clotting in your circulatory system. And coagulation is clotting. But what happens normally is you use up all your factors, your fibrinogen, and the rest when we get into it, and then you start bleeding, and then you've got big problems. We already got to erythrocytes? Okay. Erythrocytes. Red blood cells is an erythrocyte. They carry oxygen. That's what a red blood cell does. That's what it's there for. Now, one of the things that we talked about when we talked about glycolysis is that the red blood cells do not use oxidative glycolysis. It's all anaerobic. They're carrying the oxygen for the tissue. They don't want to be using it themselves. You know, it's kind of like the pizza guy eating the pizza on his way to deliver it. They need, you know, so it lacks nuclei, it lacks mitochondria. They only have about 120-day lifespan. They have hemoglobin, each molecule of hemoglobin can bind, I believe, 4 oxygen, and transferrin, and their job is to carry oxygen to the tissues. Red blood cell will be 6 to 10 microns, usually 7 to 8 microns in size, just big enough to fit through the capillaries. While they're in the capillaries, they release the oxygen and pick up carbon dioxide by diffusion. How is the gas exchanged in the capillaries? It's by diffusion. There's not an active transfer. Okay? And it's the same with glucose, the same with any other waste products through the capillaries. Not necessarily into the red cells, but it is by diffusion. These are red cells. Now, notice they're biconcave discs. When they get into the capillaries, they change shape a little bit. They have to bend sometimes to get through the capillaries. And by being biconcave, they have a lot more surface area. A lot more surface area aids in diffusion. You think diffusion is going to show up on the next test? Probably a pretty good idea.
There's nothing usable word diffusion. You don't even know what diffusion means. It's not a
Excuse me a second. I am on call. I told you right at the beginning.
Should probably just go and
I probably should just go into work, shouldn't I? Call it a night right now.
We don't want to make you late. We don't want to make anybody wait for an important class.
Are you short of funds? Get funds transferred to your account today. God, where did they get my number? Or how did they know?
like you never use that.
Yeah.
I'm sorry, was I not supposed to give it out?
No, probably not a good idea. And it'll show up. Okay, leukocytes. Leukocytes are the white blood cells. You have the granular and you have the agranular. The granular are your neutrophils, your basophils, your eosinophils. Your agranular are your monocytes and your lymphocytes. Okay, so here you have neutrophils, polymorpho nucleocytes, because you see the different shape of the nucleus. These are the ones that fight bacteria. These are the ones that fight bacteria. The eosinophils, they're called eosinophils because the stain that caused the granules is orange and the stain is eosin. It picks up the eosin. They're eosinophils. They will be seen when you have an allergic reaction. That's what you see when you have an allergic reaction. Basophils. Basophils are only about 0.1% of the blood. Normally, if you see them, you're looking at a heavy metal poisoning. And here you have a lymphocyte. Lymphocytes are the ones that produce a lot of the antibodies, a lot of the gamma globulins. They fight viral disease. Now, if a patient comes in, and I'm doing a CBC, and I really can't color on this, but you see this little blue rim, and that's kind of a pale blue? If it's out as a very dark blue, almost as dark as, say, the nucleus and the eosinophils, a very dark, very rich blue, that is what we call a reactive or an atypical lymphocyte. That lymphocyte is producing antibody. There is a viral infection someplace. A lot of the times we can diagnose a mononucleosis by simply looking at the blood smear, looking at the lymphocytes. Monocytes, again, help in the viral infection. Again, nowhere near as numerous as the lymphocytes. Platelets are cytoplasmic, bits of cytoplasmic tissue off the megakaryocytes from the bone marrow, and they are totally just used for clotting. They are the first things to react to a cut, and they form a plug. They release more clotting factors. They start the clotting cascade that eventually ends up causing the fibrinogen in the serum or plasma to become insoluble and form strands, which form the actual solid plug that stops the bleeding or plugs up a cut. Oh, and then you have the red blood cells, and we've talked about those. Platelets, they're a formed element. They have no nuclei. Very short-lived, 5 to 9 days. And they are needed to clot blood. They form the initial plug when you have a clot. is a disease called von Willebrand's. Von Willebrand's is tissue factor or clotting factor number 9. He discovered it. Oh, okay. But anyway, they cause the platelets to adhere together, and so you form a much stronger plug until the fibrinogen and the rest of the clot forms. Okay, formed elements in the blood. You have the red blood cells, you have the white cells. Red cells, biconcave, without a nucleus, 100 to 120 days, 4 to 6 million per millimeter cubed. A millimeter cubed is, a cubic centimeter is about the size of a sugar cube. A millimeter cubed is 1/1000th of that. So it's very small. So there's 4 to 6 million. Transports oxygen, carbon dioxide. White blood cells, granulocytes. Now, if you remember, the granulocytes are the ones that have the granules in the cytoplasm. Neutrophils, eosinophils, basophils. That's a granulocyte. Neutrophil. It's 50 to 60% of the white cells, phagocytic. They go around eating up bacteria. Eosinophils, they stain with the eosin stain, 1 to 3%. It helps detoxify foreign substances, secretes enzymes that dissolve clots, fights parasitic infections, and normally if you get, if you come back from Mexico with E. histolytica or something like that, a nice parasite, you'll see the eosinophils up.
Was that for allergy too?
Yes. Yes. You'll see them up in allergies. And it doesn't say that here, but that's actually where you see them mostly. Basophil, dark blue, big granules all over the top of the nucleus. It does release heparin as an anticoagulant as you start to break down clots. Because when you form a clot, at some point it's got to break down and the vein get back to normal. Agranulocytes, no granules. Last a lot longer, 100 to 300 days. Monocytes, 2 to 3 times larger than a lymph. It's phagocytic. It goes around again. It still picks up some bacteria and some viral particles. Lymphocytes, larger than the red cells, nucleated, provides immune response. And immune response is the response that your body makes to an antibody, and we will get into that. Immune response is what you use when you give a vaccine. You're introducing a foreign substance to your body. You're letting the lymphocytes and your immune system recognize that it's foreign and make an antibody against it. What you're doing is it takes a week or two or more initially. So if you've got a very serious virus, and you might not live long enough to take 2 or 3 or 4 weeks to make the antibody to it. So we give it as a vaccine in very low doses, which still does the job, but your body recognizes it as foreign, makes the antibody to it, then when the natural virus hits, it's a matter of hours to maybe a day, and your antibody titers go through the roof. That's how vaccines work. Okay, hematopoiesis, process of blood formation. What's that?
I got the anthrax vaccine when I was in the military, so does that mean that I had anthrax?
That means
in my body?
they either gave you an altered anthrax, or they gave you a killed attenuated, what they would call it is attenuated anthrax. And what would happen is it's not alive or it's not in a position to make you sick from anthrax, but it does introduce your body to anthrax so that you will form the antibodies. And then if you do get exposed to anthrax, your body's reaction will be a matter of hours to a day as opposed to days to weeks. And in days to weeks with anthrax, you're dead. So, yeah, they did give you some sort of anthrax. Leukapheresis, white blood cells, the bone marrow, lymphoid tissues, cytokine regulation, that's a regulation as to the body's regulating to what it needs through the cytokines. We'll get back to that. Red blood cells, RBC, erythropoietin. Erythropoietin is secreted by the kidneys, and it is partially in response to low blood pressure and low blood oxygen. And there's sensors in the kidneys for this, and it says we need more red blood cells. It secretes erythropoietin that gets into the bone marrow and tells the bone marrow to start releasing more red blood cells. Hepcidin is secreted by the liver. It helps regulate the iron metabolism. That's about as much as we're going to get into hepcidin. So here's the cascade. How you doing? Now this is for red blood cells. You have a stem cell. You have a hemocytoblast. If you see those in the peripheral blood, you've got one really sick cookie. To a proerythroblast, occasionally you'll see that in the blood. To an erythroblast, these are in the bone marrow. When you get your erythropoietin released, it stimulates the transformation from those to erythroblasts to a normal blast. A normal blast is what we would call a nucleated red blood cell. You will occasionally see those in the peripheral blood, mainly if somebody has been bleeding and the bone marrow is just kicking out whatever red cells it's got. Now, if you've ever seen a doctor order a reticulocyte count, we use a vital stain, a super blue, and we stain the red blood cells, and we see these in the young red blood cells. These cells haven't been in circulation long enough to get through the spleen, which picks these particles out. But we can tell by the percentage if the body is making red blood cells. If we have somebody that's anemic and there's no reticulocytes, then the idea is, hey, the bone marrow isn't kicking out blood, and we're looking at an aplastic anemia, maybe a chemical poisoning. But we do this test all the time for people that are anemic. And then eventually they mature to the point that they're erythrocytes. That if you get normal blasts or reticulocytes, by the time they hit the spleen is going to pop those particles out of the red blood cell. So you get somebody that's had a splenectomy, you see more of the nucleated and reticulocyted type particles in the blood.
When old red blood cells rupture, the released hemoglobin is ingested by macrophages. The globin chains of hemoglobin are broken down to individual amino acids that are metabolized or used to build new proteins. Iron is released from the heme of hemoglobin. The remaining structure is converted to biliverdin, which is then converted into bilirubin. Iron is transported by transferrin in the blood to various tissues for storage, or to red bone marrow for making new hemoglobin. Free bilirubin is transported by albumin in the blood to the liver. Liver cells make conjugated bilirubin, which is excreted as part of the bile into the small intestine. Intestinal bacteria convert bilirubin into bilirubin derivatives, which contribute to the color of feces. Some of the bilirubin derivatives are absorbed into the blood and excreted from the kidneys in the urine.
Got all that? Did I hear the word board? Okay, red blood cell antigens and blood typing. What type of blood is it? A, B, A, B, and O. Close. Okay. So what happens here is this was I mean, the luck was almost amazing. A guy named Landsteiner in about 1906 had 4 people in his lab, and he drew blood from all 4. And he had an A, he had a B, he had an AB, and he had an O. And that is where we got the 4 different blood types. Now today, we know, we can actually look at the chemical composition on the surface of the red blood cell. But what we're dealing with is 4 different blood types. That's your ABO system. Now your positive and negative is a different system. It's called the RH system. The RH comes from the rhesus monkey, where we first discovered them. We discovered those in about 1948 or 49. Yeah.
That's actually a question. I found out that my blood was AB negative, and
Okay.
the person explained to me, like, the negative won't matter until you have a second child, because if you have a RH positive fetus, it'll attack it and try to...
I will get into hemolytic disease in a moment. Okay.
I want to hear about what I have to do.
Take Rogaine.
Yeah.
You're going to automatically get a Rogaine. We virtually see none of it in this country anymore because every woman that is negative, RH negative, is going to get a Rogaine shot at 28 weeks. And it is so bad that we have to import the anti-D from other countries where women don't get the Rogaine. But we'll get into it. Yeah. So A has type A antigen on it, B has B antigen, AB has both, and O has neither. Okay, now this is going to get a little bit, this is a circular argument. So stay with me, it's not that bad. An antigen is a substance in the body. An antibody is a substance the blood makes against an antigen. Okay? So, if I have an antibody for hepatitis, for the hepatitis virus, at some point my body has been exposed to a hepatitis virus in order to make an antibody against it. The antigen is the hepatitis virus. The antibody is the hepatitis antibody. Whatever the body makes against the hepatitis antigen or the hepatitis virus. Okay? Hepatitis is not a naturally occurring antibody, meaning you have to be exposed to it to get your body to create it. It's not that way with the blood groups. If you have a person that is type A, they naturally have a type B antibody. So if you were to take an A person and give them B cells, whether they had been exposed to B cells or not, this antibody is going to hemolyze those cells, and you are going to be in big trouble. So it's a naturally occurring antibody. So A has a naturally occurring anti-B. B has a naturally occurring anti-A because it recognizes that A is foreign. A recognizes that B is foreign. Naturally occurring, we don't know why. Now, if you have AB, you've got both the substances on your red cells. So A is not foreign because it's on your red cells. B is not foreign because it's on your red cells. What antibodies do you have? No. You have none. O is, I'll get to the O's. So you have the substance on your cell, so your body doesn't recognize it as foreign, so you can, it won't make any antibodies. Now O, on the other hand,
So that's why there's the universal receiver?
it's a universal receiver. Yes. O, on the other hand, doesn't have A on the cells or B on the cells. So it makes both anti-A and anti-B. So when we type your blood for a doctor, for a transfusion, for a whatever, we do what we call a forward type. So we take the red blood cells and we put them in a solution of anti-A, of anti-B, and actually anti-D to look for an RH. So if they're an A and you put them in a solution of anti-A and spin them, they form a nice big clot, a nice big chunk. If they're B, when we put them in the anti-B, they form a chunk. If they're AB, we put them, the A forms a chunk, the B forms a clone. So either the A tube clots, and it's A, the B tube clots and it's B, or they both clot and it's AB, or neither of them clot because what cell doesn't have anything on it? O. That's how we type the blood. Not really, because we only use sheet, but if you want to, I can bring the types in and some typing syrup. If you want to see how it's done, we'll do a demonstration. That's totally against it. The blood I bring will be from Red Cross, so it's already been tested for the HIV and hepatitis and about 1 million other things. So, then we do a back type. So we put A cells in one, and if it's A, it doesn't clot. But if it's B, it's got anti-A, and it clots the cells. So we actually type in two different directions. We test for the antibodies on the cell surface. We test for the antibodies in the serum. And normally that works really well, unless you get somebody that is say A2 with an anti-A1. Because you have varying, you don't see it with B, but you do see it with A. So they have a type A or a type A1, or they have a type A2. If you have an A2 and they've been exposed, this is not naturally occurring, but if they've gotten blood, and the blood's generally an A1, they'll make an anti-A1. So all of a sudden your back type won't work at all. And then you just throw up your hands and give them all. But let's look at this. Transfusion reactions. Like I said, A makes anti-B, B makes anti-A, O makes both of them, AB makes neither. So in AB, if you put A blood into an AB person, nothing's going to happen because there's no anti-A. You can put B blood into an AB person. Nothing's going to happen. Okay? You can put O blood in. Nothing's going to happen. Now on the other hand, if I've got an A person and I put B blood in, they've got anti-B in their serum, and the B has B on the cells. it is going to hemolyze those cells, and you're going to have one really sick cookie who, if you don't act quickly, is going to die. B, if you put B, gave A cells to a B person, same thing. Now the O cells do not have anything on them. That's why they are the universal donor. But let me be very clear. It's O cells, not O blood. Because the plasma in O is full of anti-A and anti-B. So unless you're giving it to an O person, the anti-A and anti-B that you give in the plasma are going to attack the cells of the person you're trying to help.
Can't be whole blood, right?
It can't be whole blood. And it's very, I mean, I don't think I've given a unit of whole blood in probably 10 years, 15 years. It's just a waste of resources. So, the only time that we give whole blood, if it might be a directed donor, and I haven't seen one of those in probably 10 years now. So, that's why O is a universal donor. There's nothing on it. You can give it to anybody if they have anti-A or anti-B in their system. There's nothing on the O cells for it to attack, to recognize as foreign. You can give it to anybody, the cells, not the plasma. AB, on the other hand, you can only give AB, is a universal recipient because there's no antibodies in the serum. Because everything is recognized as self. So you can give them A blood, you can give them B blood, you can give them O blood, or AB. I don't even keep AB in my laboratory. If I get an AB person that needs blood, I give them either A or B, and since A is very common, I just give them A. I've always got plenty of it around. It's, if I give them AB, if I have AB, then I have to worry about it outdating. Because I can't give AB to an A, I can't give AB to a B, and I certainly can't give it to an O. The only person I can give it to is an AB, and I just don't see that many transfusions like that. So that I would be throwing these units away. So I don't even keep it in the laboratory. Transfusion reaction. If a person receives the wrong blood type, antibodies bind to the erythrocytes and cause agglutination. They agglutinate and then they hemolyze. They break down. And that is a nasty, nasty thing to see. I have never, in 34, 35 years now, ever seen a hemolytic transfusion reaction to a wrong blood type. Never seen it. The safeguards you take are tremendous to avoid that. I know it's happened. The guy that invented plasma that saved hundreds of thousands of lives in World War II died in a North Carolina hospital, I think, because they gave him the wrong type of blood.
Is there anything you do to once you realize you've not had blood?
Yeah, there is. But essentially, you give them the right type of blood, you give them a lot of fluids, you put them on dialysis, and you get them to a tertiary care center. Now, the only real hemolytic reaction that I've seen in the 35 years was not caused by a blood type problem. It was caused by the fact that the patient was given cefotetan, cephalosporin antibiotic. And if you look in the literature, it binds to the red blood cells, and about 2 or 3% of them, it'll cause hemolysis in certain women. So this lady comes in and she has a C-section, and part of the standard protocol is to give them cefotetan to prevent post-op infections. She goes home. After a while, it kicks in and starts hemolyzing her blood. And she comes back from the hospital a few days later saying, I'm getting weak. I've got, my urine's red. The doctor says, oh, you probably have a UTI, and gives her another big dose of cefotetan, which tends to hemolyze all the rest of the blood. And the only reason we were able to pick it up is, one, we knew we hadn't given her blood. Two, it creates the autoantibodies, so we would see it. When we do a blood type, we always do it with the patient's blood with nothing else, see if it clots on its own, and this one did. So we knew we had some sort of an autoimmune reaction going on. But this lady lived, but it wasn't by much. We ended up, because once this reaction starts, you have maybe 1 unit out of 5,000 that is actually compatible. And so, as soon as we, I remember I walked into the intensive care, and I told the hematologist, we can't handle this one. We got to get her out of here. And she went up to San Francisco, put on dialysis to remove the blood. A lot of what happens is the membranes from the red blood cells that have broken down clog the kidneys, and you go into renal failure. So they put them on dialysis to get rid of that, and she did survive, but it was really touch and go for a long time. Yeah.
When it says the case, which one do they use? Which one is better? To do with the one that is written here or the peritoneal? Which one?
Oh, you can't. You're not going to do it with peritoneal dialysis. It has to be. You have to get in, and you have to be pumping the blood right through those filters. Yeah, peritoneal dialysis will only remove fluid and electrolytes.
Is it possible, let's say, if it gets better, to start here and then move it to the one right here?
No, peritoneal dialysis is used in emergency situations if you don't have a dialysis, or in somebody that, the kidney patients that they don't have any veins after a while. That's why they go in and put a shunt in to use. But with a peritoneal dialysis, you essentially have an open wound into your peritoneum, and you're already a sick cookie. You get an infection in there, you might as well kiss it goodbye. So they do use it on occasion, but it's only in an emergency. So. Okay, transfusion if you give the wrong type of blood. Transfusion reaction. This is what we've been talking about. The antibodies, type A will agglutinate type B will agglutinate type A. So, with an anti-B and an anti-A, this is how we would type. This is an anti-B serum, this is an anti-A serum. The A blood cells, because they don't have any B substance on it, is smooth, it doesn't clot at all. With the anti-A, it clots the A cells. Exactly opposite with the B, and the AB, everything clots. Okay, RH factor, rhesus factor, that's what we call normally an anti-D, but it can be an anti-D, anti-C, anti-E. There's 3 different antibodies in the RH system. And each of these are called a system. The ABO system, the RH system, the KEL system, the MNS system. There's a bunch of them out there. RH is probably as far as we're going to go. Anti-D, we call it anti-D. And if you're D negative, you're RH negative. If you're D positive, you're RH positive. So what we were talking about earlier is a mother who is RH negative. If the baby is RH positive, sometimes the cells from the baby can cross the placenta and get into the mother's blood. So now you have an RH positive cell in an RH negative mother. The RH negative mother is her immune system is going to see that RH positive cell as foreign. Even though it's her baby, it's going to see it as foreign. And it's going to turn around and make an anti-D, like the anti-A or the anti-B. It's not naturally occurring, but it's going to happen. And there's, you have to, one cell isn't going to do it. You have to have, a certain dose, and I forget exactly what it is. I think it's like 20 cc, something like that. But this anti-D can cross the placenta. And so it crosses the placenta, it goes right back into the baby, and it starts hemolyzing the baby's cells while it's still in the uterus. That's your hemolytic disease in the newborn. The reason the baby, in the first couple of pregnancies, the uterus is still pretty thick and you don't get a lot of bleeding, and the babies are usually okay. They may come out, they may be anemic, their bilirubin might be up. You can deal with it. After the second or third baby, the walls of the uterus get thin, and you get more of this across, and a lot of times the baby will die before it can be born. And then you get a stillbirth. And this, I mean, out in the desert, I would see women coming across from Mexico, and they would have 2, maybe 3 live children, and then have lost 7, 8, or 9 children because of this. And at that point, there's really not much you can do about it. They've already got the anti-D. What we do now is, when we know the mother is D negative, we give her a substance we call Rogam. I might talk about it later, in a minute. We give her a substance you call Rogam, RHO immune globulin. We essentially give her anti-D. It ties up the antigen sites on the red cells from the baby, and if the antigen sites are tied up, the body doesn't recognize it as foreign, so it doesn't create an anti-D. We don't give enough anti-D to cause the baby problems, but we do give enough to keep the mother from developing the antibody. And that's why this erythroblastosis fetalis is virtually not seen in this country anymore. Not in women that have had their prenatal care here. Okay. Enough for now. We'll come back. We'll start with blood clotting. It's kind of a whole different thought process. And we'll start with that on Tuesday. What we're going to do tonight in the lab is we have rabbit muscle. And what we'll start off with is we'll work as tables. I will cut you little lengths of rabbit muscle. You're going to tease it apart to get down to one muscle fiber. You're going to need 4 or 5 of them. And it'll be in glycerin. And you'll put it on a microscope slide under a cover slip, and then you're going to add sodium, you're going to add ATP, you're going to add sodium plus ATP, and these are going to cause the muscle fibers to contract. So you're going to put the fiber up. I mean, is it bright out?
No, I have a cold and the light is killing me.
Okay. You're going to put them under the microscope, under a cover slip. On the edge of the cover slip, you're going to add the chemicals. You'll cause the muscles to contract. You measure the muscle before you do that, and then you look at the percent of contraction. After that, we'll give you one unknown, see what you come up with. Okay? In the meantime, I will pull one of the flexicomps out, the computer systems out, and with a couple of volunteers, we'll try to get it set up. And we'll try to look at some muscle spikes with flexation, extension, look at the coactivity and that. But that will simply be done as a demonstration. And so it shouldn't be a real late night. Let's. You want to take 2 minutes and take a break, and then we'll head over to the other lab. What's that? Does anybody want the test results? I've got them. Let me ask you something. I mean, I'm a little bit flabbergasted at the test results. When I wrote the tests, I went from 200 questions down to 65 questions. And each time I looked at a question, I said, how many times have I talked about this? Now, for instance, question, I think it's number 22. 15 out of 20 people missed that question. If your pupil is constricting, what type of system is doing this? It's the parasympathetic system. We talked about it when we talked about the autonomic nervous system. We talked about it when we talked about fight and flight. We talked about it with the sympathetic system. We talked about it with the parasympathetic system. We talked about it again when we talked about the eyeball. I could count somewhere between 7 or 10 times in 3 weeks. I brought that subject up and I said, this is parasympathetic, this is sympathetic. The eyes in fight and flight, your pupils go wide open. That's sympathetic. To have as much light in as to have the best vision possible. When the parasympathetic kicks in, they close down. I just have a question. How else can I do this? I mean, when I did it, I think I counted 7 different topics that we touched on that question on. Yet still, 3/4 of the class missed it when they saw it on the test. And I'm not trying to be facetious. I'm just trying to find a way to write the test that I'm not confusing. Yeah.
What was, do you know what the option that most of us chose was? Like, do you have any idea what we chose?
I would have to go back and look. No, it just told me how many missed. I would bet it was probably sympathetic system, which is, this is the question where, in actuality, the sympathetic system is antagonistic.
Sorry, I remember looking at that one, but I remember I didn't say parasympathetic or sympathetic. There was like a focusing or anybody know what I'm talking about?
I think one of the other options was like a motor response or something.
Yeah, but you see, a motor neuron response is not. But I mean, I don't know if at the end of each chapter I should give you a list of study questions to go over, or I mean, let me just give you the 10 test questions at the end of the chapter.
Do a take home, so I do my poli-sci class. Take homes are evil.
I've given one take home in my career, and they hated it. A 10-question test, I think the average was about 15 pages of answers for a 10-question
I'm talking
test.
like Scantron style.
Multiple choice.
I had a take-home test that was 100 questions in A&P, and we worked together in groups, and there were 6 of us, and it took us 14 hours to finish it. Not worth
They're
it.
10 times harder.
Oh, and on a take-home test, because it's, you've got the book and you got the time and you got friends to work with. Any question is fair. It's not going to be, what's CNS versus ANS. Any question is fair. But I would certainly appreciate, and we've got 2 more tests. The final is not optional. Okay?
The next one is, right?
The next one is. I have kind of inborn bias against, not taking the test, not doing your best to learn it and take the test. But that's my personal thing. On the other hand, I'm legally required to give you a final, so the final is not optional. You cannot drop the final. You have to be here to take it. I will give this to you in writing when I hit the end of the semester. If I find that the grades are really skewed down, obviously not up, but if they are skewed down, I will, at my discretion, possibly adjust the cutoff points a % or two in order to make it more of a bell curve on the grades, which means I'm grading a little bit on a curve, not the straight 90, 80, 70, 60. That will be at my discretion when I see all the grades in at the end of the semester. So you may possibly come in with an 88 and still get an 80. But I will not guarantee that. It depends. Right now, when I look at the grades, they pretty much match a bell curve of what you would expect as a distribution through a class this size. So if I see a shift in that, and I will give this to you in writing so that, because it is a little bit of a change from the syllabus. The other thing in the syllabus, because I was questioned, is the number of points. And the number of points are approximate. Your grade is going to depend on the percentage of the total, not on the actual number of points that you get. And I want to make that very clear. Because if we don't do a lab, if we get, or if we have an extra lab and I can adjust the number of points that I give you, if there's more points, then you have more opportunities to score. And almost everybody's getting, almost perfect scores on their lab. But just be very, it's not even an argument I'm going to have. Your grades, and it says in your syllabus that the total points are approximate and they can change. And they probably will. But your grade is going to depend on your percentage of the total points. Okay? Take a few minutes. If you have suggestions, I'm certainly open to them.
So are you going to go to the questionnaires within each chapter?
Let me look at that, because in all reality, the questions that I'm using on your test are very similar to the knowledge questions at the end of the chapter and the questions on the study section, the student study section, the quizzes. They're damn near identical. In all reality, and if you go through those things and you're not doing well on those questions, and this is, and I've told you this a couple of times, you need to go back to that area of the chapter and reread and refamiliarize yourself. If you answer all the questions about A, B, O, R, H, and you don't have a problem with it, don't worry about it. They're not going to be any harder than what you see there on your difficult quiz question. But use it as a template as to where to go back and study for the exam. Okay? And like I said, when I go through and I make the exam, I am mentally counting up the number of times we've talked about the subject in class. I'm not trying to make it tricky. There is online. Right in the front, there's a website, MHHE, and you go under the student edition, and and then go to the chapters. In each chapter, they'll have somewhere between 40 and 50 quiz questions. And they're extremely similar to the questions that are going to be on the test. But the idea is when you go through that, know the answers to the questions, because some of them are going to be identical. But the other is, if there's a part that you don't know, that you're coming up bad, go back and review that part. Spend your time on the stuff that you don't know. Another example is, I think 2/3 of the class missed the sodium-potassium pump. How do you return to depolarization or to polarization? It's the sodium-potassium pump. We probably hit on that a dozen times in the last 3 or 4 weeks. This is what just absolutely, and the vast majority of you missed it.
It was kind of confusing, but I didn't miss it, but I
The
almost did.
wording was kind of tricky for me on this test compared to the others, to be honest. I don't know if it was.
Yeah. It's like super wordy.
The,
I just like.
You should have said like instead of the ciliary muscles
Okay.
contracting. you should
Everybody
put
but
eyes
Carrie can have
open.
input. Okay, let's take a quick break and let's get over to the other class.
If the black part of your eye gets really tiny, what's going on?